In the name of God. In the second part, we become familiar with the components of the innate immune system and also their functions. The components of the innate immune system are epithelial barriers and some effector cells of innate immunity, such as neutrophils, mononuclear phagocytes, and natural killer cells, circulating effector proteins of innate immunity, and cytokines of the innate immune system. Epithelial barriers involve skin, gastrointestinal mucosal surface, respiratory mucosal surfaces, and these barriers produce some kind of antimicrobial peptides. Also, these barriers contain intraepithelial T lymphocytes, B1 lymphocytes, and mast cells that are located under many epithelia and in serosal cavities. These epithelial cells could act as a physical barrier to infection and could prevent the entrance of the microbes to the body. Also, these epithelial cells can produce antimicrobial peptides, and some of these peptides are defensins and catalysidins, and these peptides could act like antibiotics and could kill the microbes. And also we have some kinds of cells that are present intraepithelial, and they are intraepithelial lymphocytes. These lymphocytes, contrary to the lymphocytes of adaptive immunity, show limited diversity and has the ability of killing the microbes or infected cells. Effector cells of innate immune system include phagocytes like neutrophils and macrophages, dendritic cells, and natural killer cells. Neutrophils or polymorphonuclears are the most abundant population of circulating white blood cells. They are produced in bone marrow, and cytokines like GCSF and GM-CSF has a prominent role in production and maturation of neutrophils in bone marrow. Neutrophils could mediate the earliest phases of inflammatory responses. As you know, the first cell that enters to the site of infection from the blood circulation is neutrophil, and these neutrophils has some kind of granules. These granules contain enzymes such as lysozyme, collagenase, and elastase, and also some other kind of granules which contain microbicidal substances. Neutrophils has a nucleus with 3 to 5 lobes. Because of this structure in their nucleus, they are called polymorphonuclear cells. An adult human produces more than 10 to the power of 11 neutrophil per day, each of which circulates in blood for a few hours or days. After entering the tissue, neutrophils function only for 1 to 2 days and then they die. Because the content of their granules cannot attach to basic stains or acidic stains, they are called neutrophils. The functions of neutrophils. First function of the neutrophils is phagocytosis and destroying of microbes, especially opsonized microbes. When a microbe covered with some components like complement components or immunoglobulins, the neutrophils can recognize them better and they uptake these microbes, they phagocytose them, and after that they can destroy these microbes. The other function of the neutrophils is producing antimicrobial reagents for extracellular microbes. So, as you see in this picture, sometimes the neutrophil could uptake the microbe and destroy the microbe inside the neutrophil, and sometimes the neutrophil could degranulate their contents of the granules and kill the microbe outside the neutrophil. And also some neutrophils could produce some nets, and these nets are neutrophil extracellular traps, which compose of nucleic acids and some proteins, and as you see, the microbe could be trapped in these nets, and by this function the neutrophil kill the microbe, and also itself is dying. The other population of innate immune cells are mononuclear phagocytes. We have some circulating monocytes and some tissue-resident macrophages, and both of them belong to mononuclear phagocytes. Mononuclear phagocytes respond to microbes nearly as rapidly as neutrophils do, but macrophages survive much longer at site of inflammation. Macrophages are not terminally differentiated and can undergo cell division at an inflammatory site. Macrophages are the dominant effector cells of the later stages of innate immune responses. For example, 1 or 2 days after the infection, macrophages are more dominant compared to neutrophils at the site of infection. About the maturation of mononuclear phagocytes, we have some macrophages that are originated from fetal hematopoietic organs such as fetal liver or yolk sac, and they could differentiate to tissue resident macrophages. such as microglial cells, which are the macrophages that are reside in brain, Kupffer cells, which are macrophages in the liver, some alveolar macrophages in the lung, and some sinusoidal macrophages in the spleen. But we have other population of macrophages which are differentiated from monocytes. As you see, these macrophages are originated from hematopoietic stem cells, and these hematopoietic stem cells could produce monocyte dendritic cell precursors, and they differentiate to monoblast and then monocytes. After the infection, at the site of the inflammation, the monocytes exit from the blood circulation and enter the site of the inflammation, and there they can differentiate to macrophages. So these kind of macrophages are not resident in tissue, and they can differentiate from the monocytes. Monocytes, as you know, have a bean-shaped nucleus, and we have two different types of monocytes: classical monocytes and non-classical monocytes. Classical monocytes are the monocytes that recruit to the site of infection following the neutrophil. So these kind of monocytes are called classical monocytes. But we have the other class of monocytes which are called non-classical monocytes, and these non-classical monocytes more involved in tissue repair at later stages of, in final stages of the inflammation. Macrophages and neutrophils could phagocyte the microbe, and after the phagocytosis happen, we need a kind of killing process in the macrophage or neutrophil. We have three different mechanisms for killing of phagocytosed microbes. The first one is provided by proteolytic enzymes. These proteolytic enzymes in the phagolysosomes could lyse the microbes, and both activated macrophages and neutrophils could use this mechanism for killing the microbes. The second one is production of phagocyte is the activation of phagocyte oxidase system. Activated neutrophils and to a lesser extent macrophages convert molecular oxygen, which means O2, into reactive oxygen species and or ROS. And as you know, this ROS is toxic for the phagocytosed microbe. So this is a very important mechanism in neutrophils, activated neutrophils, and also in macrophages for killing the phagocytosed microbes. And the third mechanism is inducible nitric oxide synthase. Activated macrophages could produce reactive nitrogen intermediates, mainly nitric oxide. And this nitric oxide could be toxic for the microbe, and this nitric oxide could be produced by the action of inducible nitric oxide synthase, or INOS. So we have three different mechanisms for killing of the phagocytosed microbe. The first one is proteolytic enzymes such as elastases and cathepsin G, which are present in lysosome, and after the integration of the phagosome to lysosome, this enzyme could kill the microbes. The second one is production of reactive oxygen species by phagocyte oxidase, and the third one is the production of nitric oxide by inducible nitric oxide synthase. Here you see that a microbe could be attached, for example, to a kind of PRR, like mannose receptor on the surface of a cell, and then you see that this membrane zips up around the microbe, and we have a microbe which is ingested in a phagosome. So we have a phagosome with ingested microbe, and also we have a lysosome which is full of enzymes like cathepsin G. After the fusion of phagosome with lysosome, we have a phagolysosome, and some microbes could be killed in these lysosomes under the enzymatic activity of the lysosomes. The other mechanism is phagocyte oxidase. The second mechanism, you see here, phagocyte oxidase are present on the membrane of phagolysosomes, and they could convert the O2 to reactive oxygen species, and this reactive oxygen species inside the phagolysosome could be toxic for the microbe. And the third mechanism is mediated by INOS. INOS is an enzyme which is present in cytosol and not in cell membrane, in cytosol, and could produce nitric oxide, and this nitric oxide, like ROS, can be toxic for the microbes that are present in phagolysosomes. And here you see the respiratory burst which can be activated inside the neutrophils or macrophages and produce reactive radicals of oxygens and also other toxic components for the microbes. If we want to summarize the function of macrophages, as you see here, after the recognition of a microbe by some kinds of PRRs on the surface of the macrophages, they become activated, they produce reactive oxygen species or nitric oxide, and they kill the microbes which are phagocytosed. They can become activated and produce cytokines such as TNF, interleukin 1 and interleukin 12. And you know that these three cytokines could induce inflammation, and also they can enhance the adaptive immunity. And at later stages of the inflammation, they could produce some growth factors, some angiogenic factors, and by producing these factors they could involve in tissue remodeling after the inflammation. The third population of innate immune cells are dendritic cells or DCs. This is linking the innate immunity to adaptive immune system. They are widely distributed in lymphoid tissue, in mucosal epithelium, and also some of the dendritic cells are presented in organ parenchyma. Most of the dendritic cells, not all of them, most of them are related to the lineage of mononuclear phagocytes. Dendritic cells could be categorized to two main populations, conventional disease and plasmacytoid disease. The main function of conventional disease are capturing the antigen, processing the antigen, and display protein antigen to T lymphocyte. You see here in the picture that an antigen enters the body, and dendritic cells which are present in a tissue can uptake that antigen. After uptaking the antigen, the dendritic cell could enter the lymphatic vessels, and by afferent vessels they could enter the lymph node, and in lymph node during this process the dendritic cell become activated and could process the protein antigen. And here in lymph node, as you see, the dendritic cell could present the antigen in the form of peptides to T cell and initiate activation of T cell adaptive immune responses. So dendritic cells, conventional dendritic cells, has three main functions: capturing the antigen, processing the antigen, protein antigens, and after processing, displaying the antigen in the form of peptides to T lymphocytes. And these dendritic cells, as antigen presenting cell, you know, they can produce different types of cytokines according to the type of the pathogen, and they can lead the differentiation of different T cell responses according to the type of the response that is necessary for combating against the microbes. So dendritic cells are the main antigen presenting cell which uptake and process the antigen, protein antigens, and present them to T helper cell and induce their differentiation to different effector T helper cells like Th1, Th2, or Th17 cells. The second category of dendritic cells are plasmacytoid dendritic cells. These dendritic cells are called plasmacytoid because they are similar to plasma cells, and they are important cells in producing the cytokines like interferon alpha and interferon beta, which belong to interferon type 1 and are very important mediators of for defense against viral infections. So the role of plasmacytoid dendritic cell is uptaking the viruses and producing type 1 interferon, which has antiviral functions. The fourth population of innate immune cells are natural killer cells. Natural killer cells are a lineage of cells related to lymphocytes, although they belong to innate immune cells. The lineage of the NK cells is common with lymphocytes. NK cells constitute 5 to 15% of the mononuclear cells in the blood and spleen. They are abundant in some tissues like liver and gravid uterus, and they do not express CD3 on their surface, so they are CD3 negative. You know that CD3 is the marker of T cells, but instead they have CD56 on their surface. So if we want to distinguish NK cells by flow cytometry, they are CD3 negative, CD56 positive, and the majority of NK cells which are present in peripheral blood are CD16 positive. About the function of NK cells could recognize infected or stressed cells. NK cells respond by directly killing the infected cells, especially viral infected cells or stress cells. Also, NK cells respond by secreting inflammatory cytokine, and these NK cells are a major source of interferon gamma, and you know that interferon gamma can activate macrophages to kill ingested microbes. So they can directly kill the stressed or infected cell. They can produce cytokines such as interferon gamma, and this interferon gamma can activate other cells like macrophages to kill the ingested microbe. Here you see NK cell can recognize injured cell or virus infected cell and directly kill these cells. Also here in row B you can see that these NK cells could produce interferon gamma, and this interferon gamma can activate the macrophage, and this macrophage after activation can kill the phagocytosed microbe. So here you see that we have an interaction between NK cell and the macrophage as two components of innate immune responses. Macrophages produce interleukin 12 and activate NK cells, and in response NK cells could produce interferon gamma, and interferon gamma can activate the macrophage for killing the phagocytosed microbe. So the function of natural killer cells involve killing of infected cells, activating macrophages to destroy phagocytosed microbes. When NK cells become activated, they release the content of the granules on the surface of the target cells. These target cells could be injured cells or could be virally infected cells, and this release is provided by exocytosis of the granules, and these granules contain perforin and some granzymes. Perforin facilitates the entry of the other granule proteins such as granzyme into the cytoplasm of target cells. Perforin is released on the surface of target cell. It can induce some pore on the surface of the target cell, and after that the granzyme, which are the enzymes that initiate apoptosis of target cells, can pass through these holes, through these holes which are produced by the action of the perforin, and they could induce the apoptosis, which is a kind of cell death in the target cell which is infected by viruses or intracellular bacteria. Some tumors, especially hematopoietic tumors, are also the targets of NK cells. NK cells have two types of receptors: activating receptors and inhibitory receptors. Activating receptors can recognize the cells that have undergone stress, the cells that are infected with viruses or other intracellular microbes, or the cells that are malignantly transformed. So these kind of cells provide the ligands for activating receptors. The other receptor on the surface of the NK cells are inhibitory receptors, and the ligands of these inhibitory receptors are MHC class 1 molecules, and you know that these MHC class 1 molecules expressed on the major healthy nucleated cells. So if a cell is healthy and also a nucleated cell, it expresses MHC class 1, and this molecule could act as a ligand for inhibitory receptors of the NK cell. The fate of NK cell activation is related to the equilibrium between these activating and inhibitory receptors. Here you see if the ligands of inhibitory receptors are more than the ligands of activating receptors, like this picture, we have more inhibitory signals for NK cells, and the NK cell cannot become activated. But if the cell, and this condition is provided by healthy cells, so if the cell of the body is healthy, not a stressed cell, the NK cell cannot kill it. But if the cell become virally infected, and in this way the expression of MHC class 1 molecules decrease, you can see that we do not have the ligand for inhibitory receptor on the surface of NK cell, and the ligands of the activating receptor is provided, and the NK cell become activated. Also, in some conditions, you can see both the ligands for activating and inhibitory cells are present, but the ligands of activating receptors are more than the ligands of the inhibitory receptors, and the net result is the activation of NK cell. So if we have more activation of activating receptor, NK cell become activated. But if the cell is not a stress cell and provide a sufficient amount of MHC class I molecules, they can attach to inhibitory receptors on the surface of the NK cells, and so the NK cell cannot be activated. Here you see we have a normal autologous cell of the body, and you see that this normal cell can express MHC class I on their surface, and this MHC class I could engage inhibitory receptor of the NK cells, so we do not have the activation of NK cell. But in the second part, we have a virus infected cell. You see the expression of MHC class I on the surface of the infected cell decreased, and we do not have any MHC class I on the surface of this virus infected cell. So this cell only provide the ligand for activating receptors, so the NK cell can become activated and kill the virus infected cell. And in the third part, you see we have a stress cell with induced expression of activating ligands. Although we have the expression of MHC class I, but we have increased the amount of activating ligands on the surface of this stress cell, so provide the activation of activating receptors on the surface of the NK cells, and the net result is the activation of NK cell, and this NK cell could lyse the stressed cell. One of the mechanisms of NK cell function is antibody-dependent cell-mediated cytotoxicity, or ADCC. You remember that I said one of the receptors which are present on the majority of peripheral blood NK cells is CD16, and this CD16 is a receptor for a part of immunoglobulin G. This is a low affinity receptor for Fc part of IgG1 and IgG3 antibodies. When a microbe is covered by IgG1 or IgG3, the Fc part of this IgG can be recognized by CD16 on the surface of the NK cell, and NK cell can kill the target cell which is covered by antibody. As you see here, we have a target cell, and this target cell is covered by antibody such as IgG1 or IgG3, you see the Fab region attached to the surface of the target cell, but the FC region can attach to CD16. These parts are CD16. After this recognition, the cross-linking of these CD16 on the surface of NK cell can trigger the granulation of the NK cell, and the perforin and also granzymes are released, and so these tumor cell or stress cells can be died by apoptosis. Other components of innate immune system are cytokines. Some of the cytokines are more prominent in innate immune system. For example, cytokines that control viral infections like interferon alpha and interferon beta. Cytokines that mediate inflammation, TNF, interleukin 1 and chemokines as the cytokines that mediate the inflammation. Some cytokines could induce the proliferation and activation of NK cell, like interleukin 15 and interleukin 2. Some cytokines, as I explained before, like interferon gamma, can be produced by NK cells, and it can affect on macrophages and activate macrophages. Some cytokines like interleukin 6 could increase the production of neutrophil from the bone marrow, and also production of some proteins like C-reactive proteins. And here you see in these tables the major cytokines that belong to innate immune system.